Hello students. In the last classes we have discussed molecular pathogenesis of cancer, chemical carcinogenesis of cancer and physical carcinogenesis. Okay. Now we are going to discuss biological carcinogenesis. Okay. So the biological agents which will cause carcinoma include mainly viruses. The main thing is viruses. Viruses cause carcinogenesis. Apart from viruses, other things like parasites, fungus, bacteria may also cause carcinogenesis. So the examples include for parasites, Schistosoma hematobium, it causes squamous cell carcinoma of the urinary bladder, and Clonotis sinensis, it causes cholangiocarcinoma. So these two are the parasitoids that cause carcinogenesis. And fungus, Aspergillus flavus, which is a fungus, when it is mainly seen in stored seeds. In that storage of any seeds are there, in that Aspergillus flavus will be there. It will release a metabolite, or it will secrete a chemical called aflatoxin. So this aflatoxin, along with the HBV infection, when it is associated with HBV infection, it may cause hepatocellular carcinoma. And bacteria like H. pylori bacteria, which is commonly seen in peptic ulcer disease. So when this gastric mucosa is continuously or repeatedly exposed to this Helicobacter pylori, then it causes carcinoma like gastric carcinoma or gastric lymphoma. So this is the bacteria which how they cause this, like what are the bacteria that causes biological carcinogenesis. Mainly it is viruses. Of all these, viruses is the most thing, is the common thing which causes carcinogenesis. So here in this class we'll be studying viral oncogenesis or viral carcinogenesis. Okay, so again these were classified into two categories. First, it is those containing DNA. The viruses containing DNA are called DNA oncogenic viruses, and the viruses which contain RNA, they are called as viral oncogenic RNA oncogenic viruses. Okay, if they are containing DNA, we call it as DNA oncogenic virus. If they are containing RNA, then we call it as RNA oncogenic virus. Okay, and uh, and again these two types of viruses, they have some oncogenic characters or oncogenic proteins which code for them are there. So these three genes are GAG gene, POL gene and ENV gene. So GAG gene, it codes for group antigen. GAG gene codes for group antigen. POL gene, it codes for polymerase enzyme. ENV gene, it codes for envelope protein. So these genes will code for the respective antigens and or polymerase enzyme or proteins. So all these were the different proteins for which these genes code for. And the transmission of viral infection takes place by three routes. One is horizontal transmission. So in horizontal transmission, it contains like a direct contact. So direct contact with the patient or inhalational exposure. So through the droplets of mucus. So that comes under horizontal transmission, or direct transmission by touch, skin touch, direct contact. So all those comes under horizontal transmission. Parental route means through blood. So that is parental route of transmission. And then vertical transmission. Vertical transmission is nothing but transmission of virus from mother to infants. From mother to infants during the delivery time or during the breastfeeding. So if this kind of transmission takes place, that we call it as vertical transmission. So the transmission of virus is through three routes: horizontal, parental and vertical transmission. And then, so the mode of oncogenesis of either DNA or RNA oncogenic virus is two different pathways. So we'll be discussing in detail the mode of DNA, in both of these mode of transmission or mode of oncogenesis that we are going to study now. So first is mode of DNA viral oncogenesis. That means DNA viral oncogen, how does it cause tumors? So first is it involves, first step is replication. So whatever the virus it is there, that will replicate in the host cell, and then it undergoes lysis, breakdown, and then it will release its virions. That is replication. Second is integration. So whatever the whatever the DNA of that virus is there, that will be get integrated into the host cell DNA. So when when it enters into host cell DNA, that we call it as integration. So during the process of integration, this will result in inducing mutations. Generally, normal DNA, when it is integrated with viral DNA, during that process, it causes certain mutations. So the outcome of these mutations is neoplastic cells. So the cells will be normal cells will be converted into neoplastic cells. So for this transmission, it requires, for this transformation for normal cell into neoplastic cell, it requires virus specific T antigens. So only in the presence of virus specific T antigens, normal cells will be converted into neoplastic cells. So that will be that you can see in this picture. So this is the DNA of a virus. So if you consider this as a virus, this yellow thing is envelope of that virus, and this red one is a DNA, and this green color dot is T gene, which codes for that particular proteins. So this is the virus, and this pink one is a host cell. So this virus enters into this host. So once it enters into the host, it enters into the cytosol or cell. So it reacts with cell membrane and enters into the cytosol. So once it is in the cytosol, this is the viral DNA, and this is the envelope, this is the T gene. This is the nucleus of the host cell, and this is the DNA of the host cell. Okay. So now it gets entered into this integrates into this nucleus. So first it is in the cytosol, now it enters into the nucleus of the host cell. So once it is in the nucleus of the host cell, what does it will do? It will undergo replication. So a number of DNAs will be produced. So that we call it as replication. And then so during the step of this conversion, so after this integration into this nucleus, a few of them undergoes direct replication, and in the presence of specific T antigen, what will happen? This viral DNA incorporates into this host DNA. So this is red in color, this is blue in color, host DNA. So when in the presence of specific T antigen, this DNA portion will be incorporated into the host cell DNA. So this is the host cells part, this is the viral part. So this is how the exchange of these chromosomes will be taking place, leading to the formation of neoplastic tumors. So this is how neoplastic cells were formed. So once it is replicated, it will cause the lysis of the cell, and it will release its DNA virions into the extracellular space. So once it is in the space, again these new virions again enters into the new cell, and again they undergo the following steps: replication, integration, and again in integration, if it undergoes another step of transformation, this transformation takes place in the presence of T antigen. In the presence of T antigen, this whatever the viral DNA, host DNA undergoes mutations and will produce neoplastic cell. So this is the mode of transmission of DNA viral oncogenesis. And then mode of RNA viral oncogenesis. So RNA viruses are also called as retroviruses. Why? Because they are having this is also called as retroviruses. They contain two identical strands of RNA, two single strands of RNA, and also an enzyme called reverse transcriptase. Okay. So this reverse transcriptase is RNA dependent DNA synthetase. It acts as a strand. So this RNA transcriptase, what does it will do? It will act as a strand and helps in the formation of another DNA. Okay. So the single strand of viral DNA is then copied by DNA dependent DNA synthetase to form another strand of complementary DNA, resulting in double stranded viral DNA or provirus. So whatever the reverse transcriptase is there, that will act as a complementary strand. So against this, another strand will be produced. So in the presence of DNA dependent DNA synthetase, this RNA will be converted into DNA. So that we call it as the double stranded DNA which is formed, that we call it as provirus. Okay. So now this provirus, as it is double stranded, now it can integrate into the host cell DNA. Okay. So generally RNA cannot integrate into the host cell DNA, right? So this RNA will be converted into double stranded DNA with the help of certain enzymes called reverse transcriptase and DNA dependent synthetase. So reverse transcriptase, how does it will help? It will help as a forming as it will be visible as a complementary strand against to which this again new strand will be formed. So this is how double stranded DNA will be produced. So this double-stranded DNA of this virus is called as provirus. So now this provirus will integrate into the host cell DNA, double-stranded DNA. So during this process, again replication will be taking place and formation of neoplastic new cells will be done. So retroviruses are replication competent. The host cells which allow replication of integrated are called permissive cells. So as they're permitting it to undergo replication, so these were called as permissive cells. Non-permissive cells do not permit replication of integrated retrovirus. So non-permissible cells, they will not allow this RNA virus to enter into the host cell. And then viral replication begins after integration of the provirus into host cell genome. So viral replication will start only when this provirus, or which is double-stranded provirus, it is there when it integrates into host cell DNA, only then replication will be starting. So once replication is done, again it causes lysis of the cell and will release its provirus into the circulation or provirus into the extracellular space. And again the same steps will be continued. So this forms a particular components: virion core protein from GAG gene, reverse transcriptase from pol gene, and envelope glycoprotein from ENV gene. So this is how again a whole new virus is produced. So the three components of virus particle are then assembled at the plasma membrane of the host cell, and the virus particles is released by budding off from plasma membrane, thus completing the process of replication. So once again I'll tell you in this picture. So if you consider this as a RNA virus, which is having two single-stranded and identical RNA and an enzyme called reverse transcriptase. Okay, so this reverse transcriptase, it will help in the formation of a single-stranded complementary strand. So now it gains access into the cell and is entered into the cytosol of the cell. And once it is entered into the cytosol, so this reverse transcriptase acts as template. So against this template, another complementary strand will be produced. So double-stranded DNA or double-stranded DNA was formed. This is single-stranded. So against with the help of reverse transcriptase, it acts as a template. So another strand will be formed, forming a double-stranded DNA. So this double-stranded DNA of the virus is called as provirus. Now it is formed. So this provirus now enters into the nucleus of the host cell. So this virus also have double-stranded DNA like thing, and host cell also has double-stranded DNA, right? So now it will incorporate its DNA into the host cell DNA. So this step we call it as integration. So once it is integrated into the host cell, now it starts replicating. So during this replication, what will happen? All the new cells that were formed, that will be having this mutated DNA. So that we call it as transformed cell. So again, what does it will do? Again, now after this integration, it will start replication. So by forming certain viral components. So as all its components were broken down, again with the help of certain genes like pro gene, ENV gene, GAG gene, with the help of these three genes, again they form envelope, again they'll form RNA, and everything will be formed, and again new virus will be produced. So all these new viruses, as they will form some buds like thing, that we call it as budding, and again from these buds they will be expelled out. Again they will spread to the new cells. So this is how mode of RNA virus oncogenesis takes place. And here are some examples of viruses, DNA oncogenic viruses and their associated tumors. Human papillomavirus, polyoma virus. So these were the viruses, DNA oncogenic viruses. These cause these respective cancers like cervical cancer, skin cancer, and papilloma. Herpes virus, among this herpes virus, Epstein-Barr virus, it affects the human, causes Burkitt's lymphoma or nasopharyngeal carcinoma. Human herpes virus 8, in humans it causes Kaposi sarcoma or pleural effusion lymphoma. Adenoviruses, it affects hamsters and causes sarcomas in that. Pox viruses, it affects in rabbits and also in humans, causes myxomatosis or molluscum contagiosus or papilloma. And hepatinoviruses, it affects, that is hepatitis B virus, it affects the humans, causing hepatocellular carcinoma. So all these were the DNA oncogenic viruses, and these were the respected or associated tumors, and these were the hosts in which these viruses affect. And here is another picture showing RNA oncogenic viruses. The viruses include acute transforming viruses like Rous sarcoma virus. It affects in chickens, that is hen and cock, causing sarcomas. Leukemia sarcoma virus, it affects birds, feline, bovine, that means cows and goats, primates. Primates means monkeys, chimpanzees and all. It causes leukemia and sarcomas. And slow transforming viruses, it is seen in mouse mammary tumor virus. It is seen in daughter mice causing the breast cancer. And human T cell lymphotropic virus, HTLV. Again, in this HTLV, there are two types, HTLV1 and HTLV2. HTLV1, it causes adult T cell leukemia lymphoma, and HTLV2 causes T cell variant of hairy cell leukemia. Hepatitis C virus, it also affects in humans causing hepatocellular carcinoma. So these were the RNA oncogenic viruses. And if you see this picture, so benign like molluscum contagium, that is it is caused due to pox virus, papilloma, that is HPV virus 1, 2, 4 and 7. So it causes here. And nasopharyngeal carcinoma caused by Epstein-Barr virus, it is affected here. Burkitt's lymphoma, that is Epstein-Barr virus, it is caused in the chin region. Adult T-cell leukemia lymphoma, that is caused by HTLV1. So this is the lymphoma or leukemia lymphoma. And T cell variant hairy cell leukemia, that is HTLV2, it causes adenoid carcinoma. Hepatocellular carcinoma is caused by HBV, that is hepatitis B virus and hepatitis C virus. And B cell lymphoma, that is seen in this abdominal region. And carcinoma cervix, that is human papilloma virus 16 and 18, it causes in the cervical region. Kaposis sarcoma, it can be seen in the muscle tissue. So this is caused by HHV8 virus. So these were the different viruses and that causes associated tumors. So once again go through this. If you go once again in brief, you go through this picture. Virus enters, undergoes, once it entered into the cytosol, DNA virus enters into the nucleus, and once it is in the nucleus, it will undergo replication or it will integrate into the host cell DNA with only in the presence of specific T cell antigen and undergoes integration. And whatever the new formed cells, that are the neoplastic cells. When coming to RNA, with the help of RNA reverse transcriptase, it acts as a template, so new strand will be produced. And again, this new strand, whatever it is formed, it will form the double-stranded DNA that we call it as provirus. This provirus, whichever it is formed, it enters into the host cell DNA and undergoes integration. Once it is undergoing integration, then only it starts replicating. So after integration, it will start replicating. So these will be, when it undergoes replication, it forms mutated cells. So, and also with the help of certain genes like GAG gene, Paul gene and envelope gene, newly formed viruses, budding virions will be produced. So this is whatever the cell which is having this replicated replicated DNA with the provirus, that we call it as transformed cell. So this is how carcinogenesis takes place with the help of DNA virus and RNA viruses. So this is called as biological oncogenesis. With this, we are completing this pathogenesis of cancer. Thank you all.